cupcakes. Welcome to another video. So today we will continue our discussion on the important topics for JNU examination. I have made a previous video in which I have talked to you about the important parts from biomolecules, metabolism and catalysis. If you haven't checked that video out already, please do so. The link will be in the description and I'll link it at the I button as well. So now without much further ado, let's get right into the video. Now, after discussing that one, let us go on to discuss the important topics that can be asked from the cell biology section. So in the cell biology section, firstly, you should very importantly know about the cytoskeleton. Okay, so you should know about the actin. Okay, how is actin formed? What are the monomers for actin? How the microtubules are formed and intermediate filaments. In microtubules, you specifically need to focus on the dynamic instability. Okay, there are a lot of questions that are asked, and in this you should know about the drugs, okay, that inhibit this particular process. Okay, if any drug particularly mimics this, then definitely you should know about it. You will find detailed information very nicely given on this in Albert's, so you can refer to that one as well. Then the second one that I will like to tell you is you should know about the motor proteins, that is dynein, kinesins and myosin. Okay, now, what about it, especially the route of transport? So from where it plus n to minus n or minus to plus, which particular direction you should know. Then in the myosin, you should also know about the A bands and the, the Z bands and all that, anisotropic bands, I bands, you should know. Okay. So that is about this one. Then you should also know about the cell adhesion molecules. So which are the different cell adhesion molecules that you should be knowing. Okay. Then next one you should do is the Na plus K plus pump. A lot of questions are asked on this pump. Okay, how many Nas for how many Ks, all that is asked. Then I'll tell specially like you to focus on two organelles, that is the ER, endoplasmic reticulum, both the rough ER and the smooth ER, and the mitochondria. A lot of questions are asked on the mitochondria, including cell biology as well as in the biochemistry section also. Then next you have to do the nucleosome. Okay, so nucleosome, the dimensions, the packing, exactly you should be able to identify. And then about the telomeres, okay, the sequence, the enzyme, what is the function of this telomerase enzyme, etc. Then next important topic that I want you to do is the G proteins and the GPCRs. Okay, very important. How do they function? What are the binding sites? What is the downstream? That is important. Then you should know the functions of these enzymes, that is pKa, that is protein kinase A. What is the function of phospholipase? Okay, what is protein kinase G? What is DAG, IP3? Very, very important. Next, a lot of questions, again concentrated lot of questions on the cell cycle control. Okay, so what is the control when you go from G1 to the next phase, from G2 to M, from metaphase to anaphase? What is the APC complex? What are cyclins involved? All of this you have to do in the cell cycle control. And lastly, cancer, again very, very important. In that you should know about the proto-oncogenes. Then what are oncogenes? Then what is a tumor suppressor genes? Okay, RB53, RP53, retinoblastoma protein. All these are definitely important. Okay, and lastly, in cell biology itself, I will add you should do bar bodies. You know how to predict how many bar bodies will be there. What are bar bodies exactly? Those. Okay, so that is from cell biology. Next one will move to plants. Okay, so what is important in the plant biology? So firstly, the important is the phosphorylation. Okay, the photosystems, cyclic photosystem 1, photosystem 2, cyclic photophosphorylation, non-cyclic. Okay, so that is important. The exact photosystems, at what temperature, at what wavelength do they absorb? Such questions you have to do. The next one is the chemo osmotic hypothesis. Okay, so should do this definitely. Then next you need to do is the very simple, you will say, but there is a lot of questions that are asked, that is from plant reproduction along with the types of pollination. Okay, very easy, but so many questions have been asked from this section. Now, from where would I recommend this? This you should do from the 12th textbook. Okay, enough information is provided over there for you to answer any question that can come from this section. So definitely it will be very easier to understand also. Then you should know about the C3, C4, especially what is the first product of these cycles. Okay, they ask this question very frequently. And you should know about the CAM pathway, the CAM plant, and examples in these. Okay, they may give you they'll give you four options and tell you, tell which one of them will follow C3 or which of them follows C4. That type of questions. Then again, very important questions asked in possibly all entrance exams is on the plant hormones and their functions. Okay, so such type of questions you have to do. And lastly, from the plant section, what questions I have to tell you is based on the ABC model. Okay, so mutation in one of the genes would lead to what formation. So such type you have to do. I have a video on this, a comprehensive video in which I have even solved questions that are asked in CSIR. So if you do that particular thing, then you will be very, safe for this part. So that's about the plant. Guys, if you're still watching my video and if you haven't liked it or subscribed to my channel, please do so. That really motivates me to keep going on and on for you. Okay, so now let me tell you the next part, and that is based on genetics. Again, in genetics, there are quite some topics which you will have to cover. They are very important. Now, with genetics, what you have to do, first of all, your Mendelian genetics should be very strong. Okay, all the ratio for the dihybrid cross, for the trihybrid cross, what is a test cross, a back cross, the Mendelian characters chosen, all are very important. Again, for this 12th textbook is your easiest spot to clear all of this. So do this. Then second one, the thing that I will say is pedigree. Pedigrees are also asked in CSIR and GATE. They're very easy to solve once you get the hang of it. Again, I have made a very comprehensive video. I have solved many questions also on this. If you are interested, definitely do check out those videos as well. Then the next thing that they ask is the operons. A lot of questions, a lot means a lot of questions have come from the lac operon. Okay, and some questions on the trip operon, but majorly on the lac. They ask you detailed questions even on the type of partial diploids formed. Such questions they definitely do ask. Then next you have questions on DNA replication. So they will mostly ask you the different types of polymerase and what reaction do they catalyze. Okay, even on RNA polymerase, so which of them are sensitive to amanitin, that is also asked. So such type of questions. And in after DNA replication, the next concentrated questions come from from protein production, that is basically translation mRNA to protein. So basically the A site. Okay, then how the particular tRNA comes and binds to these sites. Then what happens on the P site, what happens on the E site, where does the first tRNA bind. All of those questions they are asking on this. So when you're doing translation, even focus on the steps, how the ribosome situates itself on the mRNA, and about the tRNA also you should know in detail. Okay, then next one that you should do is about the DNA binding motifs. Okay, specifically in DNA binding motif, what they ask you is on the zinc finger. Okay, and the second type of question that they ask you is on the homeodomain. So again, very important to do. Then finally, after this, what is next important is you should be able to tell what is the function of the chaperone. Not much details, okay, just what is the function. Then next question is based on ubiquitin and ubiquitinylation. So what are ubiquitin? What is the function of ubiquitinylation? That you should know. Nothing in much details, just about this. Then this is everybody's favorite question, types of mutation. Okay, then what is the Ames test and what are the repair mechanisms? Okay, not very detailed questions asked, just go through it once and you will be able to answer. Then next, what type of questions? Very important. Lately, I've seen many questions on this is epigenetics. So they will ask you what is epigenetics and what are the common epigenetic modifications done. So what are the, like, there is acetylation, then lysine groups are added. Such type of questions, then they become very important to do. So that one on epigenetics also. Okay, so that is the eighth most important topic. And finally, the last one that you should do is on transposons. Okay, so the different types of transposons, at least you should know the name. See, nobody will ask you the exact mechanism of how the entire thing goes down, but definitely there are questions that are asked on, identify which of these are the transposons. A last topic that I would like to add, that you don't actually specifically need to know very detailed, but they ask you analytical question on this, and that is the Mendelssohn-Stahl experiment. They ask you, like, if there were these many molecules, so tell me after how many rounds you'll have, how many bands, where the bands will be. Okay, such type of questions they ask from this genetics section. Then for rDNA technology, okay, recombinant DNA technology, there aren't much question asked. The usual questions is, what are the cloning vectors, the elements that should be present in a cloning vector, and you should know a little bit basic about the expression vector also, how both of the cloning and the expression vector are different. And then very important, a lot of questions again are asked on the cDNA. So what is cDNA library? This type of questions, simple basic level is asked. Then next section that is asked is on viruses and prokaryotes. Now there can be a lot of different questions that are asked, but I have tried to sum it down to smaller ones that are usually repeated a lot. And that is the nutritional type of organisms, again asked in GATE also, like chemoorganolithotrophs. So what do they use? Those type. There are different types. So based on the carbon source, the electron source, and the light source, energy source, they have basically divided the different types. Okay, so you should know about each of those. Then next one, they ask about the bacterial toxins. So you should do that section well. Then regarding phylogeny, and in this basically you should know about the 16S rRNA and about the mitochondrial DNA, what is the significance, according to evolution. Then very important, numericals on growth. Okay, so there are a few formulas, just know those and you'll be able to solve this. Then in this, again, an important section to ask questions is on nitrification. Very important. Okay, so what are the enzymes? What are the different processes that go on? Pertaining to bacterial, obviously they are asked. Then specifically one virus that I found a lot of questions is on hepatitis. So all the different A, B, C, D, okay, all the hepatitis you should do well. And lastly, the question that they ask is on agrobacterium. In that, more specific, if I have to tell you, it is the virgins. Okay, so questions on this have been asked in the JNUEE paper. Fine. So that is all about the viruses and prokaryotes. Next on immunology. Now immunology isn't as vast as any other topics here. The topics are very confined, and in that they ask you detailed questions. Like in antibodies, there are majority of the questions. If I say 80% of the question asked in the paper are based on antibodies. Okay, in that you should know about the monoclonal antibodies, about the hybridoma technology, very clear about the concept of the light chains, heavy chains, variable region, constant region, the abundance of different antibodies. Again, I have a video on this one. If you are interested to know more, you can definitely look at this video as well. So don't forget to do antibodies very thoroughly. Then about the MHCs also. So the class 1 MHC, class 2 MHC, what cells are involved, what type of differences, the similarities, again, very important to do. Okay. Then the next topic, very important to do, is based on vaccines. So the types of vaccines, that type of questions are asked. And you have questions on the characteristics of certain cells. Okay, specifically their focus is on the NK cell and the macrophages. Okay, so this, pay a little more attention while studying. And the other type of question they ask is on hypersensitivity. So types of hypersensitivity, what is the tuberculin test, okay, which hypersensitivity class it falls under, just take a look. And lastly, a new type which I've not seen much but I'm still mentioning is on the map kinase pathway. Just look through that. So that is about the immunology section. I know this is a lot, but I have really tried to include most of it that you can cover in these coming days. And finally, you have on the physiology. Okay, so two more sections to go. One is the ecology and one is in physiology. So now in physiology, my sincere recommendation to you is go through the 12th and 11th textbooks for your particular. You can go through NCRT because that's one standard book. Every information is definitely mentioned over there. It is really great to look at. Okay, so all the things and specifically which chapters you should look at. So in control and coordination, okay, in that what are the different centers, okay, which is the sleep center, where is it, where is the anger center, where is the satiety center, all those questions are asked. Then next one, what they ask you is based on the hormones. So in that only there is a continuation on the hormones. So what are the function of those and what goes on if any of them are imbalanced. Then the very other important, very important, star raised to infinity important is reproduction. Okay, everything you need to know about the uh, spermatogenesis, oogenesis, about the contraceptives, about fertilization, the acrosome reaction, about zona pellucida, all of those are very important. Then in excretion and osmoregulation, the important part, if I have to tell you in this, is basically the urine filtration, the formation of urine, the Bowman's capsule loop of Henle. Those questions are more definitely asked, and the regulation mechanism, so involving the RAS system and the aldosterone, etc. Those type is very important from this one. Okay, and there was one question on Hox genes also. uh, it is something that is optional. If you know it, you do it, otherwise don't spend much time on this because it's not that it is asked every year. Okay, so that is about the physiology. Then next one is ecology and the ecosystem. So not again many questions, but very important that is asked is the Hardy-Weinberg. They definitely ask questions on Hardy-Weinberg, the numerical types. So again, I have a video on this. Do check it out. I have tried to include everything. I've solved some CSIR questions also in that video. So it will definitely be helpful to you. I'll link it again. So do check it out on Hardy-Weinberg. Then the next type is they ask you about the characteristics of an ecosystem. Okay, so such type of questions are asked. And finally, in terms of evolution, they ask you the types of speciation. Okay, then the definitions like what is a genetic drift, what is genetic shift, all of those are commonly asked. And finally, the last ending questions you will basically see are more of the numerical types. And in that, the major questions that come, as I was telling you, from thermodynamics, that is on enthalpy, entropy. So again, I have a video, I'll link it out, go check it out. And then there are questions on pH. Okay, very important questions on pH. They ask you questions on the dissociation constant. Okay, and finally, most commonly asked questions is based on the restriction digestion. I cannot tell you how important it is. Every time they do have questions, and these are not very easy for you to solve without practicing before. So definitely you do need practice for these type of questions. If you want me to make any videos from these topics that you think is very important before the exam, do let me know. And guys, join my Telegram channel. I have shared a lot of the past year's questions there, so if you have to solve them, definitely that would be helpful. And that's it from me for this video. I'll see you in my next one. Bye.